I just want to say, being here, sitting here, walking around, squeezing my way around, listening in and asking people questions, I'm so proud. I'm really, really proud of my college. I'm proud of the faculty who are so dedicated to providing authentic research experiences to our students, whether that be in the classroom, side by side in the lab, out in the field, or over mathematical equations or a computer. We all understand how important it is when you learn science and mathematics that you are doing science and mathematics. We know that research experiences are tied to higher retention rates and higher graduation rates for our students. So I'm very, very proud. I'm even more proud of the students. When you walk around this room and you see the level of work, the quality of research, the intricacy of the questions that they're addressing with their work, it is amazing every single year to be in here and see this. I don't know another institution where we have students, undergraduate students, working at the level that they're working here. And I think it says a lot for our students. So you work very hard, you take a lot of classes, you find time for this, you find time for this in the evenings, you find time for this at the weekends, you find time for this in the summer. So I'm very, very proud of all of you. Beyond that, so I'm excited to be here, I'm proud. I had 3 things that I had to remember to say: excited, proud, and grateful. So the last one is grateful. I can't see him, but I want to extend my thanks to Associate Dean Yin-Fan Ma, who's our Associate Dean for Research. He is a tireless advocate for research, for faculty research and for student research. If you haven't met him, you know you haven't met him, because if you have, you really know you have. He has so much energy. He's kind of like bouncing around up here in the roof space right now. He took this program on, he's put an awful lot of effort into it, and he's the reason why we're going to need a bigger room next year. I'd also like to thank the staff of the College of Natural Sciences and Mathematics, especially Rosemary Castillo, who is the main organizer of this event. But all the rest of them are here this evening. So Naomi Robinson is here, Matt Young is here, our safety manager Brittany Anderson-Steele is here, so if you have any issues about safety, she'll answer your questions. Laura Emerson, our resource analyst, is here. So all the faculty are here. Thank you for coming and for being here and supporting this, and thank you to our community members and family members who have also come out this evening to support our students and appreciate the work they do. So just a big thank you to everybody. You all deserve a round of applause. And with that, I'm going to hand off really quickly to the provost for a couple of short words, and then we'll have one announcement, and then I'll let you get back to it.
Thank you, Lisa. So a couple of short words, so I don't have very long time. Big, long, speech to give. I'm very excited about it. Let me just reiterate thanks. I've had lots of experiences at universities. When I was an undergrad, I was lots of different biology majors. I was a physics major for a while. I ended up as an economics major. But I did work in a lab there, and I was looking through the program, and somebody out here is trying to break apart DNA and identify it. We were doing something like that relative to tuna protein. And loved it. Personally, though, the impact of students being involved in research, I feel with my son. My son was a chemistry major, sort of liked it, sort of liked doing research, and then he started working in a lab with a professor, and it changed everything he thought about what he was going to do with the rest of his life and how he was going to do it. He's now a graduate student studying to get his PhD in chemistry. So let me say a handful of thank yous. First, thank you to the faculty that are doing this work with the students. It is unbelievably and incredibly impactful. Your students appreciate it, and we appreciate it tremendously. So a big round of applause one more time for our And thank you for the students. Lisa said she wants to get a bigger room. I want to get a smaller room. Smaller room with more people so that the energy that's in here can be captured even better and bring them together. You walk around, thank you for putting yourself out there. It's frightening to present your work and to reveal what you've been working on to people and to talk to them about it and have questions asked. So thank you for your courage and your resilience and your work. Thank you for telling me about the Pacific Herring that I just heard about. Wonderful project. Last but certainly not least, how many family members or friends or supporters are in the room? Let's have a big round of applause for that. You've You've put your trust in us to take care of your friends, your family members, your children, whomever is here, and we deeply appreciate it, and we take that trust very seriously. I hope that what you're seeing in this room is the care and the love that we have for your students that you put in our care, and you've seen the results that come from that. So thank you all for being here. Enjoy the rest of the evening. Wait till next year when it's even smaller and there's more.
Thank you, Provost Perez. I'm just going to invite Professor Ron Coleman up here for a quick announcement. He's the director of the Center for Undergraduate Research here on campus.
Thank you. Hi, Ron Coleman. It's actually the Student Research Center, and it's in the second floor of the library. And why I'm here is because this isn't the end. You know you're doing work, you're going to keep doing work, and you've got more opportunities to tell people about what it is that you do. You've got 2 opportunities coming up. The Student Research Center hosts the Fall Poster Forum, which occurs on November 4th, and you can still sign up. There's flyers at the front door. You can sign up until tomorrow. Every poster that's here should be there, because here you're representing your lab, you're representing your department. On Monday, November 4th, you're representing your college. So let's show them what NSM can do. Okay, it's university-wide. We want to have a huge presence from NSM. We already have a large one. We want to have even larger. And that's not it. There's one other thing coming up. In the spring is the Student Research Symposium, and that's your chance to talk, orally present your work. We want to see every one of you out there telling the world all the wonderful things that you do. Congratulations.
Okay, thank you for taking a moment to listen to us. Get on back to your research and presenting your work, and let the energy come back into the room. Thank you.
My poster, we are looking at past burning in California by Native Americans. And one of the reasons we're looking at this is it may potentially help give some guidance to future fire management strategies, because clearly we've had an issue with fires becoming more intense and more frequent in California. And our rationale is that when Europeans showed up, they thought they were looking at a pristine environment here in California, actually all across the United States. And there is growing support that in fact the landscape had been heavily manipulated by the Native Americans. Burning is a cultural practice that they've done for a long time. In this instance, we think that they had been doing it to manipulate the landscape, to improve game, and acorns are a big part of their diet in California, so that's a potential reason for doing it. And what we did was I am counting charcoal sediments. That it comes that you stick a core down in the ground, pull out some dirt, and it's a very long history of it, like over thousands of years. So I'm counting the charcoal that's in that sediment and identifying spikes in really heavy fire activity. And what we are going to do is we're going to date that, and we're going to compare it to what we know the climate was and what we should be seeing vegetation-wise, what kind of trees, grass. If we see that there is a significant discrepancy between what should be there and what isn't, that's a really good indication that there was some human activity involved. So what we did was we went out, we were at several different sites in Yosemite National Park, because this area has been occupied by Native Americans for 10,000 years. There is a lot of archaeological evidence we'll be able to compare to see if we're getting the same feedback from them.
Hi, my name is Brandon Thomas, and my project was on track reconstruction and performance analysis of the pixel detector at Atlas. So this summer, I had the opportunity of doing research at CERN, which is the European Organization for Nuclear Research, which is a collaboration of scientists and engineers that work together to expand our understanding of high-energy physics. One of the primary ways they do this is via the Large Hadron Collider, or the LHC, which performs proton-proton collisions at a center of mass energy of 13 tera-electron volts. In order to analyze these collisions, the researchers use detectors, one of the largest and most important of which is Atlas. And Atlas itself is comprised of a couple different detection mediums. The closest to the beamline, that proton-proton collision, is the pixel detector. Now, when these particles collide, resultant particles will pass through the pixel detector and free up electrons, leaving a detectable hit on that silicon. In order to comb through this data, researchers will use neural networks that help them reconstruct the tracks and disregard things that they might not want to look at. But sometimes these neural networks can be wrong, and we want to look at those processes in which the neural networks misidentified clusters. So our team was tasked with writing a program that would help aid in analysis of some of these hits and verify their credibility after they get run through this neural network. So what we had to do was we had to take data that was given to us from the Atlas detector. We had to read it into this program so that we could play around with it, basically. We had to simulate the first two layers of geometry of the pixel detector, which are the IBL and the B layer. We reconstructed these tracks using track properties like the momentum, the charge, the primary vertex. We had to merge all of this into one rotatable window, which we used for the event visualization environment. So we took data from the Atlas detector and we filled these Lego plots, or three-dimensional histograms, with the charge distribution and the rows and the columns so that we could visualize the charge distribution across these pixel detector modules. And then we had to merge all of those different components into one graphical user interface so that someone could actually use this for themselves. So this project, what we're able to do with it is we're able to reconstruct these tracks, and we're able to read out the momentum and the first two layers that they passed through in the pixel detector. We're able to visualize these charge distributions across individual modules. Charge distributions that are from these split hits, these special cases, are going to be marked with different colors. And all of this is changeable within a easy to access parameters file. So here's a little visual of where we're at with the current project iteration. And for this project, it's basically wrapped up. But future work, we're interested in pursuing different high-energy physics analysis. The next thing on the table is going to be studying long-flying B hadrons through the insertable B layer of Atlas.
Hello, my name is Kayla Jenkins, and I did research over the summer with Dr. Amy Wagner in the geology department. My research has created the map of oxygen isotope values. So it all started this summer when I took an amazing research opportunity from STEM Seas for students to have experience among research vessels. And so you can see that from San Diego to Alaska, I collected these water samples over a 10-day cruise, and then when I came back, I analyzed them using the liquid water isotope analyzer here. So those values that I got were delta 18 oxygen isotope values that were processed, and then I put them on a map using latitude and longitude. What you observe is more depletion happening in the delta 18 oxygen ratio as you go more north. In comparison from the map that I created to a map from nasa.gov, we can see that the range is actually larger, and that's due to the fact that there's not a lot of samples being taken in this area. And so because I took so many samples over the summer, I was able to make a higher resolution map. The importance of this higher resolution map is for paleoceanographers to better understand modern climate and also past climates, to see what's actually occurring during climate change.
My name is Matt Martinez, and I have been working in Dr. Altman's lab studying Alzheimer's disease. We have been working with brain endothelial cells, which is what you see in these three pictures up here. And the red that you see represents cell damage, and the blue is the nucleus of the cell. In this image here, this group of cells was treated with the harmful amyloid beta molecule, as well as what we think to be the beneficial good molecule called sphingosine 1-phosphate, or S1P for short. And we hypothesized that the S1P molecule would protect cells, these endothelial brain cells, from the harmful effects that the amyloid beta molecule can have on these cells. And what our results show through this picture is that because there is less red, which indicates cell damage, it tells us that the S1P molecule is actually protecting brain cells from this harmful amyloid beta protein that is in a overabundance in people with Alzheimer's disease. And this is great news because that tells us that S1P may one day be used as a new treatment for Alzheimer's disease because of the benefit that it has on these brain cells.
I've never seen such a wide range of amazing undergraduate various sciences in one place, so sophisticated and interesting and informed and dynamic. I've never seen such a thing.
My name's Jacqueline Stein, and I work in Dr. Kimberly Mulligan's lab, and we were investigating how the environmental toxin BPA affects the neurodevelopment of fruit flies with the FMR1 mutation, one of the most common single gene causes of the autism spectrum disorder. One of the things we were looking at is how the neural stem cells proliferate in the brain, where in normal flies we see a certain amount of proliferation in the brain, but with FMR1 flies we see an increase in that proliferation rate. So more cells end up being in the brain overall. And what we wanted to see is how that proliferation rate could be impacted by an environmental chemical BPA. So what we would do is introduce the flies to the chemical in their food, and they would ingest it, and we would take the first generation of that fly line and dissect their brains and then visualize it with certain antibodies to determine how many cells are dividing in that brain. So we would stain these stain for neural stem cells, while this antibody stains for cells that are just dividing in general. So we would overlap that image and create a 3D render in our Amaris bitplane software, where we were able to actually quantify with a subjective, without any bias, a quantitative count of how many cells are actually dividing in the brain. And what we ended up finding was that we saw a significant decrease in the wild type flies, the normal flies. We saw a significant decrease in the proliferation rate, which is something we were not expecting. Our hypothesis initially was to figure out, it was initially that we saw thinking we were going to see an increase in the proliferation, coinciding with the BPA, coinciding with the neurodevelopmental disorder. However, we ended up seeing the opposite effect, which is something we weren't expecting to see, but we are going to be looking at in future directions. We're going to be looking at certain analyses like an RNA sequencing to determine the molecular, how to understand how molecularly this disorder and BPA are interacting. And we are going to be also investigating the different analogs of BPA. So things that are BPA free are being replaced with chemical analogs that are chemically similar but are different enough to be classified as different chemicals. So we're going to be seeing if the chemicals that are being replaced have any similar properties in the neurodevelopmental effects.
I'm Sierra Rack, and Grass Valley is my study area. Now, the geology here is pretty complex, so we have some questions there, and we also have a question about why there's so much gold here. Now, historically, Grass Valley is very productive in its gold distribution. So we have it's the actual location of Empire Mine. Now, Empire Mine was the most productive mine in all of California's history. And so we have the question, why is there so much gold here, and does it have anything to do with its geologic formation? Now the rocks here are typical of an ophiolite. You know, an ophiolite is an ocean crust that's in place onto a continent by some tectonic process. So here is your typical stratigraphic column of what a mid-ocean ridge ophiolite would look like. These are your typical rocks. This is very simplified. Not many ophiolites look like this. But here in our rocks at Grass Valley, we can clearly see that we have mantle material. Now here we have ultramafic rocks from the mantle, and our rocks are serpentinized, so we call them serpentinites. Just above that, we see gabbros. So we have this teal in our map is our gabbros, and just above that we have diabase. Now in this case, in your general ophiolite, you would have sheeted dikes, but now here is the stratigraphic column of Grass Valley. Now it kind of looks like a mess from here, but you do see some of these dikes. None of them are sheeted. So these sheeted dikes are actually formed by an extensional process that's typical of your mid-ocean ridge at the formation of ocean crust. So you would have these vertical intrusions of diabase that's forming ocean crust. Now we do not see any sheeted dikes in our Grass Valley rocks, so that's a little something curious about our rocks. Now, but just on top of that diabase, we have our typical volcanics. Now here in our simplified ophiolite, that would be translated to your pillow basalts that you would see at the surface of the ocean. So we have our volcanics, our diabase, our gabbros, and our ultramafics. So we have an ophiolite. But now there's been something that's happened with these rocks, and you can tell that they've been altered in some way. So what we did to get to the bottom of this, and to get to the bottom of why there's so much gold here, is to go out into the field and collect rocks. So this is kind of one of my field areas. It's a very beautiful field area that I get to spend time in. And then this here is one of my gabbros. Now you can tell that there's some foliation here. These rocks have been smushed or smashed by some compression. So we can't just make determinations of what process these rocks have been through by field observation. We actually have to take a look at our rocks very, very small scale. So what we do is we take these rocks and we slice them up to 30 microns thick. So what we see in our gabbros, and it's typical of most gabbros, is we'll see clinoproxene. So that's a mineral, and this clinoproxene paired with an albite, in this case we have plagioclase, that's typical of your gabbro. But here at the very rim of my clinoproxene, you'll see that there's a hornblende kind of growing on the very edge of it. So we have hornblende throughout these rocks, which is a relatively medium pressure, medium temperature rock. So these rocks are not just forming at a very low temperature. There's some kind of a high pressure signature there. Now this is the same thin section, a different portion, but we see the same signature. Here I have a different sample. This is going to be our diabase. Now this is a diabase porphyry, so we have small phenocrysts throughout this rock. Now we might have some hornblendes here, but we certainly have some chlorite, which is a relatively low temperature, low pressure alteration of these rocks in a diabase matrix. So we have some kind of a medium temperature, medium pressure mixed up with some low pressure, low temperature in a relatively nearby area. So what's going on with these? Well, we couldn't just tell from just the thin sections. So we took a look at the geochemistry of our rocks. So we sent out our rocks to a lab, and what this lab does is they crush up the rocks and get a whole rock composition, so an elemental composition of my rocks. What elements are here? Are there rare earth elements, trace elements, and what have these rocks been through? So in addition to that geochemistry, we also took our rocks to UC Davis to use their microprobe. And so what this microprobe does is it shoots a beam of electrons at a specific mineral of my choosing, and what it'll give back is an elemental composition of my rocks as well. But what this specifically will give back is it will help me identify these minerals. So I don't just have to identify them just by sight. I can actually get an elemental composition. So this is the data that I get back. So this data I can use to confirm the minerals that I have, and I can also use the Al2O3 and TiO2 content to determine and plot them on a temperature pressure line. So here, the majority of my rocks are relatively low temperature, low pressure, but I do have some scatter here. So, and it's mostly in my gabbros. So there is kind of a medium temperature, medium pressure here that I'm seeing based on the data given back from the microprobe data. But as well as the microprobe data, I'm giving back geochemistry. And so what I get back is I get similar to this data, I get back a certain amount of elements within my rocks. So this is called a spider diagram. And this spider diagram is actually used to normalize these rocks to a standard. In this case, my standard would be a mid-ocean ridge basalt. So some of the basalts that we're seeing over here in your standard ophiolite. Since mine are so weird, we're going to get some geochemistry to see what signatures these elements will give back. So what we see normally, our normal line would be right here at 1. But these are all over the place, right? So what we see here consistently throughout all of my rocks is a depletion in niobium and an enrichment in thorium. So that is very typical of, and it's a signature of a subduction zone. So not only were my rocks formed at a low temperature, low pressure environment, they were also subjected to some subduction. So what we're thinking is that these rocks were formed in this kind of an environment. So they may have been formed in an island arc environment and then later on subjected to some subduction. Now, to answer the question of why there's gold here, what we did is we included a trace element in our geochemistry data. And so we actually had to look at the parts per billion. In economic geology, typically we use parts per million, but even at parts per billion, we see hardly any gold. So we can conclude that the ophiolite actually did not contribute to the gold distribution of the area. Exactly. Yep. So these late plutonic activity were probably what contributed to the gold distribution in the area. So Empire Mine actually drills down just about just shy of a mile deep into the surface. So this mine gets really, really close up to this pluton. So what we have is a very hot pluton, and it's coming through and cooking all the rocks. It's carrying along gold in those hydrothermal fluids, and then that gold is mineralizing, and it has nothing to do with the rocks surrounding. So because that gold is not found in our rocks.
Hello, I'm Lisa Hammersley. I'm the interim dean in the College of Natural Sciences and Mathematics, and this is the College of Natural Sciences and Mathematics Student Research Symposium. So as you can see, we have a very, very busy room full of students, undergraduates and graduates, presenting their research through posters. We have their families here, their professors here, and members of the community who are supportive of our students doing research. It's really exciting.
This is a very compelling meeting with central new findings research coming out from an undergraduate level with high-level professors and laboratories brought together with the new Science Center under the Ernest Shannon complex. So this is very exciting because it puts the meat into the structure of the new science architecture here.
Working with is Klebsiella oxytoca, and basically our experiment is trying to figure out the effects that nitrate plays on K. oxytoca when it's grown in oxygen conditions that mimic the colon, the human colon. We perform growth curve assays at these different oxygen concentrations with varying amounts of nitrate availability to see if there is a growth advantage given to K. oxytoca, and it turns out that there in fact is increased growth when it is cultured in oxygen environments that mimic the colon. We also found out that when cultured in anoxic environments at varying concentrations of nitrate, that there's an increase in nitrate reductase enzyme activity, which also suggests that perhaps this bacteria is utilizing nitrate respiration as a means to colonize the human colon.
Posters on mouse nutritional yeast, and we're growing them in the lab, and we're trying to optimize these growth, and hopefully we will decide which culturing methods will be optimal, so that, because usually in the lot of literature there's a lot of studies that are on bacteria, and especially for fungi they're difficult to grow because they can take up to 14 days. So we're trying to increase or more like decrease that growth days, and yeah.